Hello and welcome to this masterclass series on how to sequence full-length plasmids in your own lab. My name is Anna Maria Niewiadomska, and I'm the Market Segment Manager for Public Health and Infectious Disease Microbiology at Oxford Nanopore Technologies. In the first part of this masterclass, I'll introduce you to a new way to sequence your entire plasmid before my colleague Ellie takes you through the technical process of full-length plasmid sequencing. So if you've been following this series from the beginning, then by now you should have gone through the different steps of preparing, sequencing, and analyzing your genomic material. In this masterclass, however, we're going to take all of that information and focus on a specific application, and that is going to be full-length plasmid sequencing with Oxford Nanopore. So by the time we're done with this class, you'll know how nanopore full-length plasmid sequencing differs from legacy insert-only sequencing. We'll also go through how to prepare your DNA, how to assemble your sequencing library, and how to analyze your data through the Epitome software platform. And finally, we'll also show you how you can learn more through our online resources. So let's get started with a general introduction to plasmid sequencing. First of all, what are plasmids? Plasmids are typically small, circular DNA molecules, although they can sometimes also be linear, and they exist naturally in bacteria. However, they can also be artificially engineered, and then they can be used as critical tools for genetics and biotechnology. Plasmid constructs that have been engineered now are the backbone of molecular biology, and they serve as fundamental input material with many different uses, all the way from basic research to industrial applications, gene therapy, vaccine development, genetic engineering, and much, much more. They can be used to enable fundamental studies into gene function, regulation, expression, as well as for large-scale production of proteins of interest. Plasmids can also serve as vectors for gene manipulation, mRNA vaccines, and various other applications that contribute to both scientific advancements in basic research and industrial manufacturing processes. And so, because plasmid constructs are fundamentally important for downstream experiments and applications, scientists often employ various quality control methods to verify that they indeed have the correct assembly of their plasmid, and so that they can identify any potential errors or mutations before they use them in further experiments. And so over time, Sanger sequencing has been treated as the gold standard for sequence verification and accuracy. But in practice and in reality, it can miss or obscure a lot of important biological information, especially when it comes to plasmid, gene therapy, or vaccine vector work. And so in this slide, we're going to take a look at a couple of examples of how this happens. On the top left panel, we can see overlapping peaks, and this suggests that we have contamination in our sample. And this can often happen when we've picked more than one colony or we've contaminated our sample prep for our plasmids somehow. And so this can be very difficult to resolve and deconvolute. On the top right, we see that the sequence terminates abruptly. And this is usually a sign that there is some kind of secondary structure in our plasmids that can be caused by things like hairpin loops or GC-rich regions that the polymerase has difficulty navigating. On the bottom left, we see that the sequence becomes unreadable after a long stretch of homopolymers. And this can happen because the polymerase slips after a long stretch of mononucleotides. And finally, on the bottom right, we see that the signal downstream can be quite mixed. And this is usually due to a polymorphic region, either through contamination or mutation or a real mixed sequence. So we know that Sanger sequencing has several other limitations and undesirable features, especially when it comes to plasmid sequencing, because it can only read up to a few hundred base pairs. We're generally limited to only looking at the insert in a plasmid. And it also requires primers upstream and downstream of the inserts that bind to the template DNA during the sequencing reaction. So if you're processing or outsourcing large numbers of these samples, this could accumulate to a relatively high cost. With Oxford Nanopore sequencing, on the other hand, we look at the entire plasmid, including the backbone, and this verification allows for a very thorough assessment of the entire plasmid construct, providing a comprehensive overview of not just the inserts, but the backbone sequence, as well as the structure, and any potentially unexpected modifications that might have happened during the plasmid cloning process. So let's take a quick look and compare what insert-only Sanger sequencing misses compared to Nanopore full-length plasmid sequencing. With Sanger, we don't really have any insights into the backbone, and this can mean that we have mutations in promoters or gene selection, um, open reading frames, for example. With Nanopore sequencing, we can see the entire backbone. You also avoid the need for primers and complicated primer walking if you have a larger insert. With any length nanopore reads, you can cover the entire plasmid, the entire insert with no problem. Sanger sequencing also has difficulty resolving repetitive regions, dimers, and deletions, as we saw in the previous slides. This, again, is not an issue for nanopore sequencing because we're not synthesizing anything, because we're sequencing native DNA. We can just go through the entire reads without any issues. And this also means that we can work across a large range of plasmid sizes, even to the point of going through large bacterial artificial chromosomes, something that's not possible with Sanger sequencing. And when you send off your samples for Sanger sequencing, you usually have to send them to a core sequencing provider. And this means that can take a lot of time. And it also means that you no longer have control over your data. And with nanopore sequencing, if you do this in-house, you have a lot more control and your data remains secure. So we've seen how legacy DNA sequencing methods like Sanger typically only verify a partial region of the plasmid construct. And we also know that this can result in failed experiments, unnecessary troubleshooting, and increased costs. So what's the alternative? As you'll hear in the remainder of this session, the nanopore-based workflow is easier, it's faster, and it's going to provide you more complete information to verify your entire construct and ensure robust quality control. With our complete plasmid sequencing workflow, you'll be able to generate highly accurate sequencing information for your entire plasmid construct. This will enable rapid and even same-day turnaround times at a very competitive price. It'll also provide a simple point-and-click data analysis report. Again, don't have to be a bioinformatician to be able to analyze your own data. You can just drag and drop it into our software and get a complete report at the end of the process. And this can also be facilitated in your own lab, really ensuring that your data stays secure and you have control over it. And so in the past few years, we've seen whole plasmid verification with nanopore-based sequencing really take off and become a very popular application in the scientific community. And there's many reasons for this, but some of the most common feedback that we receive is really the advantages of speed and simplicity of this workflow, and how this workflow has really streamlined cloning processes. Users are incredibly happy that they no longer have to perform test digests, colony PCRs, multiple mini preps, designing primers for primer walking. And we see a lot of enthusiasm, especially with users sequencing plasmids for gene therapy, where they often have adeno-associated virus inverted terminal repeats that can be very problematic for Sanger sequencing. And the other aspect that we see users really enthusiastic about is the increased data resolution. They really like being able to see their entire plasmid and having an annotated plasmid map that tells them that their promoters, their multiple cloning sites, and their selection genes are all intact and as they should be. And so with that, I'll now turn you over to Ellie, who's going to walk you through the details of how to get started with plasmid sequencing.
Hello, and thanks, Anna Maria. I'm Ellie Bull, and I'm a technical application scientist at Oxford Nanopore. Let's go through the plasmid sequencing workflow step by step. Here's an overview of the simple end-to-end plasmid sequencing workflow. The workflow can be divided into 4 stages. First is standard plasmid preparation, including picking colonies, culture of these in growth medium, and extraction of the plasmid DNA. Next, the extracted DNA is prepared for sequencing using the Oxford Nanopore rapid barcoding kit. Then it's time for nanopore sequencing. And finally, analysis of the sequencing data using the clone validation epitome workflow. If you're sequencing 96 samples in multiplex, you can expect results including detailed interactive reports and annotated consensus sequences approximately 12 hours from extraction of the DNA. We will go through each of these stages in more detail in the coming slides. Before we go through the method, I'd like to highlight that the entire plasmid sequencing workflow is available fully automated on Elysian. Elysian is our standalone benchtop device incorporating an integrated touchscreen, nanopore sequencing device, compute, and other necessary equipment such as a thermocycler. Following extraction off the device, the streamlined plasmid workflow minimizes hands-on time by covering everything from library preparation through to data analysis, including optional flow cell washing. Let's begin with the wet lab section of the protocol, from extraction to library preparation. After choosing your colonies and culturing them overnight, the next thing to do is perform a DNA extraction step. For this, we recommend using the QIAGEN Plasmid Plus MIDI kit, which enables the extraction of reliable quantities of high-quality DNA. You'll require 50 nanograms of DNA per sample to take forward into library preparation. As there's no limit to nanopore read length, there's no need to fragment your samples here. We also don't need to linearize circular plasmid DNA, as this step will take place during library preparation. To prepare your extracted plasmid DNA for sequencing, we recommend the rapid barcoding kit. The kit comes in 2 versions containing 24 or 96 barcodes respectively, so you can multiplex at the scale you need. These PCR-free kits use a transposase to cleave the DNA, which will linearize circular plasmids and attach barcodes in a single rapid step. The barcoded DNA samples are then pulled and a cleanup is performed using spray beads. Next, sequencing adapters are added via rapid attachment chemistry. You'll hear much more about this in the Library Prep Masterclass in this series, but as a quick reminder, the sequencing adapters feature a bound motor enzyme, which controls the translocation of DNA through the nanopore during sequencing. The adapted DNA library is then loaded onto a flow cell for sequencing. As there is no amplification during this step, the protocol only takes an hour for the preparation of 96 barcoded samples. As a quick note, if you're working with linear plasmids, we instead recommend using our native barcoding kit, which uses a ligation-based approach for PCR-free library prep. After your libraries are prepared, it's time for nanopore sequencing. As a recap, there are 2 layers of scalability to Oxford Nanopore sequencing, and the first is the choice of flow cell. Our end-to-end plasmid sequencing workflow supports the use of MinION flow cells. A MinION flow cell is capable of generating tens of gigabases of data, making it perfect for sequencing multiple small genomes. Up to 96 plasmids can be sequenced using one of these flow cells. In contrast, PromethION flow cells are capable of generating 100 plus gigabases, enough data for high coverage large genome assemblies. For higher throughput needs, you can choose to optimize your plasmid sequencing workflow for use with higher output PromethION flow cells. Here we'll proceed with our standard protocol and look at the sequencing on a MinION flow cell. The second layer of scalability is your choice of nanopore sequencing device. MinION flow cells are compatible with the portable Mark 1D, a compatible compact device designed with smaller scale experiments in mind. The sequencer can be run from a computer or laptop for on-demand sequencing wherever it is needed. For higher throughput needs, we recommend the flexible GridION device, which features powerful onboard compute and is capable of running up to 5 MinION flow cells independently. Once you have generated sufficient data from your plasmid samples, you also have the option to stop the sequencing run, wash the flow cell, and reuse it for a fresh sample. This is performed using the flow cell wash kit, which uses a nuclease that digests 99.9% of the library. You can then choose to move straight onto a new library and starting another sequencing run, or prepare the flow cell for storage, ready for another run in the future. This is an optional step, but is a simple way to maximize the throughput you get from a single flow cell. The flow cell wash kit comes in two sizes, enabling up to 6 or 48 washes. Sequencing kits for this workflow are compatible with both MinION and GridION devices, and all devices are run using the same sequencing software, which is what we'll talk through next. Now on to data analysis. Minnow is the sequencing software used on all devices. It allows real-time sequencing, base calling, and demultiplexing of your data, meaning that in the case of plasmid sequencing, after 12 hours, you have all the files necessary for downstream analysis. A standout feature of the software is the ability to monitor the run in real time. Runs can be stopped and resumed as needed. For a detailed demonstration of how to set up your sequencing run in Minnow, perform QC, and more, take a look at the masterclass, How to Analyze Your Oxford Nanopore Sequencing Data. For the next stage in analyzing your data, you'll use Epitome. Epitome is Oxford Nanopore's fully supported desktop application for nanopore sequencing analysis. It is a beautiful desktop application with an intuitive interface, and it provides a range of pre-configured workflows for all levels of expertise, including one focused on plasmids, the clone validation workflow. The Epitome platform provides intuitive and interactive reports and standard output files. Epitome workflows can be run locally or in the cloud and can be accessed through the point-and-click graphical interface, or for more advanced users from the command line. Here's how Minnow and Epitome deliver streamlined analysis of your plasmids. First, your full-length barcoded plasmids are sequenced, base called, and demultiplexed in Minnow. There are 3 base calling modes available to select from, and for this application, we recommend the high accuracy or hack mode. The data will be output as FASTQ or BAM files. Next up is analysis with Epitome clone validation workflow. To run workflow, you will need to provide your FASTQ or BAM files generated by Minnow. If you are sequencing multiple samples, then a sample sheet containing sample details is required. If desired, you can add extra columns, allowing you to add different reference sequences per barcode. Additional information about the inputs and optional parameters can be found in the workflow documentation. The clone validation workflow integrates a number of best practice tools for complete plasmid assembly and annotation into an easy-to-use analysis pipeline, including Fly or Canu for assembly, Tricycler for circularizing and refining the assembly, Medaka for sequencing polishing, and Planetate for annotation. Here is an example of a report generated through the clone validation workflow. The workflow will generate a report detailing the results of the assembled plasmid sequences, including read length and quality statistics, dot plots to reveal repeats and repetitive regions, and an annotated map of the construct for each sample. If insert primers and reference sequences are included, then additional information, including coverage and variant statistics, are reported for both the insert and backbone of the plasmid. A Medaka polished consensus FASTA sequence for each plasmid can also be downloaded for use in further downstream processes. If you're interested, an example report can be found in the workflow section of the website. Oxford Nanopore has released a freely available dataset from 96 synthetic plasmids designed to test and validate plasmid sequencing and assembly workflows using the method we have discussed today. The dataset includes plasmids ranging from 2 to 7 kb in size of varying complexities. For example, trimeric and hexameric inserts, which are often challenging or not possible to resolve using legacy plasmid sequencing techniques. The dot plot on the right-hand side confirms successful resolution of repeat sequences, which are challenging to accurately capture using short-read sequencing methods. The dataset contains POD5 signal data, along with both hack and SUP base calls. Reference information for both the plasmids and their inserts is also available. Scan the QR code on this slide if you wish to find out more. Feel free to download the dataset and explore the data and analysis pipeline for yourselves. Now we have reached the end of the sample to answer workflow. To recap, here is a list of the things you'll need to sequence your plasmids. To prepare your library for sequencing and downstream analysis, you can choose from either the 24 or 96-plex rapid barcoding kits. You'll need MinION flow cells for sequencing on your MinION or GridION device. You can also optionally purchase a wash kit, enabling the flow cell to be reused for another library. I'd like to leave you with some online resources where you can find out more about plasmid sequencing. Here are some online resources where you can find out more. Take a look by scanning the QR codes. For a summary introducing each step of the plasmid sequencing protocol, you can view our workflow overview here on the left. You'll also find a full plasmid sequencing protocol we've discussed today in the documentation section of the website. Scan the QR code here to access the protocol directly. To find out more about the many applications for Oxford Nanopore sequencing, view our applications web pages. And finally, to view webinars, white papers, brochures, and more, you can visit our resource centre. To summarize, the Oxford Nanopore plasmid sequencing workflow allows you to QC your entire plasmid, not just the insert. This includes resolution of dimers, deletions, and repetitive regions which may be missed by legacy methods. The streamlined method delivers quick results with no need to order additional primers, rapid library prep, and fast data analysis. The Epitome clone validation workflow delivers a complete, fully annotated plasmids from a simple, easy-to-use interface. And importantly, you can sequence on demand in your own lab at the scale you need, making sure that your data remains secure. Thank you very much for joining us today, and we hope you found this masterclass useful.